Well, good afternoon or good morning, depending on where you are right now. I'm Nilo Azad from Johns Hopkins, and I am very happy to introduce Dr. Guang Peng, who is a faculty member at MD Anderson for our talk today, really looking at ARID1A in cholangiocarcinoma and biliary tract cancers. Dr. Peng is a consummate physician scientist, initially doing her medical training in China and then getting her PhD at the University of South Carolina, after which time she joined the MD Anderson faculty in the Division of Cancer Prevention. Today, we're very lucky to be able to hear from her regarding her work and how this really important pathway might be meaningful for our patients. So thank you very much and welcome, Dr. Peng. Sorry for the delay, everybody.
Okay, yeah. Let me see. Okay, I'm sharing my slides. Okay, hold on. Sorry about that. Let me go open my slides. Let me see. Can you see my slides?
Not yet.
Not yet, okay. Where's my slideshow? Okay. Hello? Can you see my slides?
Yes, we can.
Okay.
Perfect.
Okay. Thank you so much for giving me the opportunity to present my work with the ARID1A. So ARID1A is very important in the cholangiocarcinoma, but in my talk, as what we did in my lab, actually we focus on the gene, we focus on the pathway instead of focus on the cancer type, because I'm basically a research scientist, focus on study genomic instability. But now, by working with the clinicians in MD Anderson, we do want to extend all our research, our observation beyond the current scope linked to the cholangiocarcinoma biology and also the treatment. So today, my talk is more on the general function of Swiss-Neve ARID1A complex in control tumor development, and also the more important things, since it's highly mutated in the cancer patient, the goal is really to functionize cancer genomic data, and we can develop clinical applications. So here, as we know, Swiss-Neve is a multi-unit complex. So it's really simplified in this cartoon. We have this core subunit, and we also have variable subunits, as you can see here. The core subunits contain ATPase activity that is used by the complex to do the chromatin remodeling. And the variable subunit is controlled Swiss-Neve complex specificity. For example, it's going to give the complex the context when and where this complex is going to perform the cellular function. So basically, Swiss-Neve chromatin remodeling complex is utilized by the cells to relax highly condensed chromatin, because the DNA is not a really fully exposed double strand. It is wrapped around the histone molecule to form a highly condensed chromatin structure. This highly condensed chromatin structure really become a barrier for the access of protein to the DNA. This protein, including transcription factors, including replication factors, and of course, the DNA repair factors. So Swiss-Neve will utilize ATP hydrolysis energy to relax highly condensed chromatin structure, which allows the access of these factors, including transcription factor, replication factor, DNA repair factor, to the DNA and to perform different cellular functions. It is a really important molecular complex because it's called evolutionarily conserved, because it's important biological function, what is the clinical relevance of SWI/SNF, it's really highlighted until the next generation sequencing technology comes into the field, and it is identified the clinical relevance of SWI/SNF complex in the human cancer. So here is a simplified heat map to indicate it. There's different subunits of SWI/SNF, and here is the ARID1A and also ARID1B, PBM1. Those are highly mutated in specific cancer type. Particularly in the ARID1A, it was originally identified in the ovarian clear cell ovarian cancer and endometrial associated ovarian cancer. The mutation rate is really high, and also in the gastric cancer, bladder cancer, and also cholangiocarcinoma, as you see in my next slides. So this is the mutation, overall mutation rate compared to other subunits. At that time, when the next generation sequence data published about ARID1A in Science in the New England Journal of Medicine, I just started my lab and I was looking for what could be a good question for me to address when I started my own lab. So I noticed the high frequent mutation rate of ARID1A in human cancers, and not many studies has been done for this emerging tumor suppressor. So in our lab, what we did is we start with the Cancer Genome Atlas tumor data. This is contains 32 cancer types, and with more than 8,000 individual tumors. So we did specifically look at the ARID1A mutation rate. So here is, I mentioned to you, this is the subtype of ovarian cancer, this stomach cancer, this bladder cancer, and this is cholangiocarcinoma. So it is really reach above 15%. So it's a major tumor suppressor in a variety of human cancers. And later, the histochemistry analysis showed 70% of ARID1A mutant tumors lose ARID1A expression due to nonsense mutation or frame shift mutation. So this mutation spectrum also give a hint ARID1A could be a tumor suppressor. And the more data was coming from the mouse model studies. Those included colon cancer, ovarian cancer, and also in the liver cancer. A variety of ARID1A knockout mouse model demonstrated it is a bona fide tumor suppressor. So based on all these studies, there's two key questions we want to address. How does ARID1A function as a tumor suppressor? And can we identify therapeutic strategies targeting ARID1A mutant tumors? These are the two essential questions we started our ARID1A project. So in order to do that, the logical thinking is we want to identify what are the proteins associated or maybe intact with ARID1A, because the interacting protein or maybe the binding pattern of ARID1A usually will give us a hint which pathway this gene may function. So we did the mass spec analysis. So we have this ARID1A as a bait. We put down all the proteins interacting with ARID1A. Among all these proteins, there's one interesting protein showed up. It's called the mismatch repair protein 2. So here are our immunoprecipitation to validate the mass spec analysis. Indeed, ARID1A intact with MS2 protein. Beyond to identify the interaction, we went a little bit depth about the molecular interaction domain between the ARID1A and the MS2. As you can see here, when we're mapping the domain, it generates a series of mutation of ARID1A constructs. This is the full length, and this indicates the small deletion. So we want to use this series deletion construct to map which region is important for the ARID1A and the MS2 interaction. The rationale to do this is because ARID1A point mutations does occur through the whole protein region. As you can see here, the protein is about 250 kD, so it's a very big protein. And to identify this interaction region may help us to identify which patient-derived point mutation in ARID1A may have functional significance. So what we did, as you can see here, this is all the different deletion constructs. we transfect into the cells, and we use the constructive pull down MSH2 to see which one lose the binding capability. And significantly, we see on the more toward the C-terminal region, that's the 1,600 to 1,700, 1,700 to 1,800. These two, about 200 amino acid, seems very important for these two proteins binding to each other. Based on this result, we pick the patient-derived mutations from the TCGA to test whether the mutations close to this region is going to affect protein-protein interaction. Indeed, as we can see here, this point mutation does show reduced binding, but this point mutation does not have the effect on the binding. So this also gives us very important evidence to show the point mutation in the ARID1A may have different functional significance, and the ARID1A mutant tumors may not function exactly the same compared to loss of the function due to their specific mutation region. So having finished this, we went on to ask what is the biological impact of ARID1A through the interaction with the MSH2. So first, I want to introduce a very simplified cartoon just to show you the function of MSH2. So MSH2 is a very important protein in the mismatch repair. This is definitely a simplified pathway for the mismatch repair. So usually you have two things can be fixed by the mismatch repair. One is the base mismatch, just as the name indicated, and also mismatch repair can fix very small deletions and insertions during the replication. Both of these pathways helped us to maintain the genomic stability, prevent introduced mutations. So basically, MSH2 is a key molecule in both sub-pathways. As you can see here, it's involved with MSH2-6. They form a heterodimer, and it involves a lot of nucleus and a series of proteins to correct the mismatch of the base and also small deletion and insertions. The clinical significance of mismatch repair pathway is highlighted by the genetic disease called Lynch syndrome. That is a cancer predisposing disease, and with increased mutation burden and also microsatellite instability. The increased mutation rate is more has a higher tendency to the Cto T mutations, as I indicated here, because the biochemistry structure, and also mismatch repair protein deficiency or mutations also occur in the somatic cancers, including colon cancer, endometrial cancer, and gastric cancer. They also have this microsatellite instability feature. So because we know ARID1A interact with mismatch repair protein 2, then naturally we want to ask whether ARID1A control mutability through regulating mismatch repair. To study the mismatch repair capacity, we do need an assay. So in this assay, we have a plasmid. This is GG mismatch. It's kind of artificially introduced a mismatch. This plasmid generates green fluorescent, and we transfect this plasmid into the cells. If the cells have a good mismatch repair capacity, it will correct this mismatch and transfer it to the GC match. That will generate orange fluorescent. So by use the flow cytometry, we can clearly quantitate the mismatch repair capacity in cells. So here is the control we did. We have colon cancer cell lines. Those cell lines have well-established mismatch repair deficiency. As you can see, this is our HCT116 cells. These cells does not have expression of MH1. That is another essential gene for the mismatch repair. And if these cells reconstituted with a small fragment of chromosome 3 and 5, you will see the mismatch repair capacity was significantly enhanced. And also, this is the correlation study of the cell lines. This is the colon cancer cell lines with the microsatellite instability feature. And these cell lines are the cell lines has a mismatch repair stability feature, and this correlated with our MMR repair assay. So from this, we are confident we have an assay that can quantitate mismatch repair capacity in the cells. So the question now is whether ARID1A deficiency associated with the impaired mismatch repair capacity. To do this, we do have a variety of cell lines. We want to demonstrate whether this pathway regulation is cell type specific or cancer type specific, or it's more general regardless of cancer type or cancer cell lines. We have shRNA to knock down ARID1A in these individual cell lines, and use these cell lines, we use our mismatch repair assay. As you can see, the red bar, that's our control plasmid. So basically, with the control plasmid, we didn't see any defect. But when we transfect with our MMR reporter plasmid, we do see ARID1A knockdown significantly reduced repair capacity. This demonstrated the causative effect of loss of ARID1A on impaired mismatch repair. So next, because MS2 is a central molecular conduct mismatch repair, our next question is to see whether ARID1A deficient is gonna affect MS2 function since these two proteins binds together. What we used is the immunofluorescence staining. We synchronize the cells in the early S phase, and also we synchronize the cells in the G2M phase. Because mismatch repair is a repair pathway coupled with DNA replication. So in the early S phase, as you can see, MS2 will form this nuclear foci, like little dots in the nucleus. This is the replication center, and this is active mismatch repair is going to occur. And also we do see ARID1A form this little foci. They do have a certain overlapping, but not completely. It is also consistent with ARID1A's other function in the transcription. So it's not 100% correlated, but we do have some correlation. And the interesting thing is this is ARID1A knockdown cells. We do see clearly the MS2 foci formation was reduced. And in the G2M phase, this is not the highly DNA replication phase. The mismatch repair protein 2 form more homogeneous staining. You don't see this foci. And ARID1A knockdown did not really change the expression or maybe the nuclear localization of MSH2 protein. So from this assay, we demonstrated ARID1A is required for the localization of MS2 protein to the replication center, or maybe coupled with the mismatch repair process. So the next question is, since we know ARID1A control MS2 function, controls mismatch repair capacity, then does ARID1A deficiency increase mutability? So to do this, we went back to the TCGA data with across 32 cancer types. Among all these cancer types with the stars we labeled, those are more significantly have the difference because the ARID1A mutation case allows us to do the statistical analysis, achieve the statistical significance. So among these, you can see this is our top 3 one, and the cholangio at this analysis, we didn't see too much difference. And the things, based on the mutation stratification, we see the trend like a little bit increase, but the sample size didn't allow us to achieve the statistical significance. And basically, as you can see here, is more dramatic in the stomach cancer. The blue represents ARID1A mutant tumor. It has an overall increased mutation burden. So this is overall mutation, collect all the nucleotide change. And here we use the analysis to correlate it with ARID1A protein level and also ARID1A mRNA level with the overall mutation burden. The result is consistent with the mutation stratification. So basically, if the tumor has a low expression of ARID1A, either at mRNA level or at protein level, we do see the increased mutation burden. And here is more specific analysis on the different substitution of base pair. So as you can see, this C2G shift is predominant in ARID1A mutant tumors. This is a wild type tumor. That's the percentage of Cto T mutations. And this is ARID1A mutant tumors. And here you can see that's the increased percentage of Cto T mutation in these cancer types. And because these are the overall, we have sufficient cancer patient numbers and also sufficient mutated tumor numbers allow us to do the statistical analysis, because ARID1A, although is highly mutated in the cholangiocarcinoma, but the overall cases number is not as big as these cancer types we showed here. So where we conduct our analysis with the ARID1A and the mismatch repair deficient tumors, there's definitely the landmark approval of FDA to use mismatch repair deficiency as the first cancer marker for the advanced tumor to the immunocheckpoint blockade. So the idea is the mismatch repair caused the overall increased mutation burden. It increased probably the neoantigen presentation, which activate T cell, cytotoxic T cell mediated adaptive immune response, and that will potentiate the immunocheckpoint blockade efficacy in the treatment. So we identified ARID1A intact with mismatch repair protein 2 and then regulate mismatch repair pathway and control the mutability in the cells. The question is, does ARID1A deficient tumors exhibit immunogenic phenotype? So what we did is we used the ID-8, this is syngeneic model, as our model system to test ARID1A deficient tumors sensitivity to immunocheckpoint blockade. So we choose this model. So first of all, because it is ovarian cancer model, although it's not the clear cell ovarian cancer, it's the major one. But this cancer model does form very consistent tumors and relatively aggressive. We have also topical injection into the ovary and also the IP injection. Both models are the established and well accepted models to test immunotherapy response. So here, this is the tumor injected in the ovary, form the tumor, and this is a normal ovary. And here is the tumors formed in the abdominal cavity and all in the peritoneal cavity. And this is the white little knots, that's the tumors. So what we did is we used the CRISPR-Cas9 to knock out ARID1A in the parental cell lines, and we inject into the mouse. Definitely we see ARID1A function as a tumor suppressor because knockdown ARID1A increased the tumor size and the tumor becomes more aggressive. We harvest the tumor and we conduct the whole exome sequence. We did observe the overall mutation load was increased in our knockdown tumors compared to the parental tumors. And here we use the IHC staining, the multiplex staining, to stain the CD8 marker, that's the cytotoxic T cell marker, and also the immunocheckpoint protein expressed in the tumors, the PD-L1. So we do see both CD8 positive cells increased and also the PD-L1 expression increased. So just notably, we didn't see ARID1A knockdown increase the PD-L1 expression in vitro culture, but this happened in tumors. So we suspect this is more like a adaptive response from the tumors because the increased cytotoxic T cells activate immunocheckpoint by increased expression of PD-L1. So this allows us to test whether we can use the immunocheckpoint blockade to treat ARID1A deficient tumors. So in addition to the orthotopical model we also used the IP model. It is very consistent with the orthotopical model, as you can see from the luciferase reading. The tumor were much more aggressive, and we see the increased CD8 and the PD-L1. We tested this model in addition to the orthotopical model is because of technical challenge to have a consistent cohort for the treatment. So we thought that this IP model could be very efficient for the treatment test in addition to the biological analysis. So to relevant our tumor analysis from the mouse model to the human situation, we did use the TCGA data to see whether ARID1A low expression tumors may have increased the TIL score. This TIL indicated the tumor infiltrated lymphocytes. This score was published in the previous papers. So we used their method to have this gene signature to indicate the TIL infiltration. As you can see here, by these two different methods published from two independent groups, we do see very consistent result. The low ARID1A expression tumors has a higher TIL score. That means it has more immunoinfiltration phenotype in the tumors. That is consistent with what we observed from the tumor analysis from the mouse. So the mouse model gave us a causative effect link, and this is result give us clinical relevance and human relevance. So here is the IHC staining. In addition to the TCGA data, use the transcriptome data. We did check the protein level in the ovarian tumors with the ARID1A deficient and the ARID1A high. We did see this increase the CD8 infiltration and also increase the PD-L1 expression. So here is the result of our treatment. As we expected, the ARID1A mutant or ARID1A knockdown tumors responds better to the immunocheckpoint blockade anti-PD-L1 antibody. So we have the mice, we inject the tumors. 1 week later, we start our treatment. This treatment only lasts for 3 weeks, twice a week. Then by the week 4, we stop the treatment. So this is day 7 when we inject tumor before the treatment. We do see the growth of the tumor. And this is day 21. And you can see this is ARID1A tumor without treatment. It is showing more aggressive growth of the tumor compared to the parental cells. And also this is the treatment with the anti-PD-L1 antibody. We do see significantly reduce the tumor burden in these mice. This is the quantitative data. So to demonstrate the therapeutic efficacy, we conducted a survival assay in addition to the tumor growth measurement. For the survival assay, we have 2 treatment strategy. The first one is we start treatment at week 1 and last for 3 weeks. And in the second treatment, we keep treating the mice until it reaches the animal protocol allowed survival. Because these mice develop ascites, so we have to sacrifice the mice when it's reached the end point. Not really the mice died, it's because the ascetics and the weight above the threshold that is allowed from the animal protocol. What we did observe is this is the ARID1A knockdown tumors. The mice survive was significantly shortened because of the aggressive tumor. And this light blue was the mice with ARID1A deficient tumor that is treated with the anti-PD-L1. So extend the life survival. But in the parental cells, these tumors were not very sensitive to the anti-PD-L1 treatment. So the treatment did not significantly improve the survival. And this may also consistent. A lot of patients in the clinical treated with the immunocheckpoint blockade did not have a clinical benefit. And more interestingly, if we prolonged our treatment, we do see there's further improved survival. So here is the ARID1A knockout tumor, the mice bearing the knockout tumor. The survival was worse than the parental. But if we keep treating the mice can survive much better than 3 weeks of treatment. So this further confirmed therapeutic benefits obtained in ARID1A deficient tumors. So we are very happy to see this result because it's kind of logically expected since the mismatch repair deficient tumors showed a better response to the immunocheckpoint blockade. But when you see our mice data, you will clearly see this is mice 1, this is another mice. They have very dramatically different survival outcome. And given these mice are really homogeneous population, and we inject very homogeneous tumor cells at the same time and treat at the same time, we already see this heterogeneous response in the immunotherapy using the anti-PD-L1. So our next question is, how can we improve the therapeutic efficacy of immune checkpoint blockade in ARID1A-deficient tumors? So this is also consistent with recent reports. Not all mismatch repair deficient tumors respond to immunocheckpoint blockade to the similar satisfying clinical response. So they do have response and do not have response, although they may, in group, they are better than the parental. I mean, the knockout without treatment. So to solve this question, we went back to the TCGA data of the endometrial cancer because this cancer do have a high mutation rate. So it allowed us to have a statistically significant result. ARID1A wild type tumor are the red tumor, ARID1A mutant tumor are the blue tumor. So you can see as I showed here. What we did is this time we're not using the transcriptome data. We're focusing on the RPPA data. That's the reverse phase protein array data. The advantage of the protein data, it is because it's a protein data, it's more directly linked to the protein function compared to the transcriptome data. But the disadvantage is we only have about 200 some proteins available for our analysis. But interestingly, when we screen the proteins significantly associated with the ARID1A mutation status, there's one protein coming to our attention, that is CHEK2. So CHEK2 is a checkpoint protein 2. Our lab is focused on the DNA damage. So this is definitely one of the star DNA damage checkpoint proteins. It is working with the ATM signaling axle. So what we observe is we observe the increase of CHEK2 expression in ARID1A mutant tumors. So it's more like toward to the red side. You see, this is the blue, red indicated tumors with ARID1A deficiency mutant, and it's turned out to be red color. That means upregulation. So basically, I show you also a simplified cartoon for the DNA damage response pathway. In the wild type cells, if you have a DNA damage, you have two major pathways. One is called ATM signaling, one is called ATR signaling. So ATM signaling is more in the G1 phase, and the ATR signaling is more toward S phase and the G2M phase. And the ATM signaling is more related to the double strand activation, and the ATR is more sensitive to the single strand activation. But these two pathways are not completely separated, as I show here. They have extensive crosstalk. For example, ATM, ATR share more than 700 common subjects because these are phosphorylation signaling cascade. ATM, ATR are both kinase. So in general, ATM activate CHEK2 pathway and the ATR activate CHEK1 pathway. So CHEK1 is very important in the S phase and the G2M phase, and the CHEK2 is more linked to the G1 phase. But also, of course, CHEK1, CHEK2 are the kinases as well, and they can be cross-activated by the ATM, ATR signaling. And also CHEK1, CHEK2 share the common substrates as well. So there's extensive crosstalk, but in general, it's defined ATM, CHEK2 signaling and the ATR CHEK1 signaling cooperate together to maintain DNA damage checkpoint activation. The checkpoint activation is more related to the cell cycle arrest. And here in ARID1A mutant cells, it has very unique changes in the DNA damage response signaling. So our previous data identified the ATR signaling was deficient in the ARID1A mutant tumors is because lost ARID1A caused the impaired activation of ATR kinase activity. So that is why the signaling pathway was kind of deregulated in the ARID1A mutant cells. And from our RPPA analysis, we see the Cto protein level was upregulated in the ARID1A mutant cells. So it's very likely this upregulation represents an adaptive or maybe compensatory response of these ARID1A mutant cells because they have defective ATR signaling. So they may need to turn on the ATM Cto signaling pathway to maintain DNA damage checkpoint. So once you have DNA damage, Check1, Check2 activate the DNA damage checkpoint, so it arrests the cell cycle and gives cells time to repair DNA and allow cells to survive. If the DNA damage checkpoint is not activated properly or repair is not completed properly, it will lead to the cell death. So the efficiency of these two pathways cooperating together is essential for the cell survival in response to DNA damage. So to confirm the TCGA analysis, what we did is, this is the quantitative data from the heat map I showed 2 slides ago. And this is our xenograft tumors we obtained from the parental tumors and also ARID1A deficient tumors. We did see this phospho Cto was activated. So the phosphorylation Cto is not just indicate the Cto protein level increase, it's also activated, the pathway is activated. That's the kinase activity is activated. And also from the clinical specimens, we validated this indeed happened in the ARID1A patient which lost ARID1A expression. So this is the case 2. We did see this patient has reduced ARID1A expression, but has increased the Cto expression. And in the patient sample, we did see this weak activation of phospho Cto. This is overall Cto. This is some activation of phospho Cto. So since we know ARID1A deficient cause the Cto upregulation, so we wanted to first to test whether ARID1A loss will increase the Cto expression. So here is the correlative data. In the ovarian cancer cell lines, we are very easily to identify the cell lines with ARID1A expression, and also cell lines do not have ARID1A expression or maybe have a reduced ARID1A expression. So in these cell lines, we do see ARID1A deficient cell lines had increased the Cto expression. And here is a more causative relationship. So this cell line does not have ARID1A expression. So we put back the Teton system to re-express the ARID1A in these cells. We did see this gonna reduce the Cto expression. So Cto is a relatively stable protein, but we did see if we introduce more ARID1A expression, it repress Cto expression. And the question is, how does ARID1A knockdown increase the Cto expression? So here we show the data. We check the RT-PCR. There's no change on the mRNA level, to our surprise, because ARID1A is supposed to be more related to the transcription regulation. But in the Cto expression case, it's not through the transcription. Then we check the protein stability by checking the ubiquination of the Cto. So that is the ubiquination assay we did. As you can see, when we overexpression the Cto construct here, and this is ARID1A wild type, this is ARID1A deficient cells. In the ARID1A deficient cells, we do see there's a reduced ubiquitination. As you can see, this is a polyubiquitination dye compared to this one. This reduced polyubiquitination may lead to the increased Cto stability because it reduced the degradation. Then the question is, how does ARID1A knockdown affect Cto ubiquitination? That is further mechanism kind of question. So to cut the story short, what we identified is we identified this CHEK2 has an E3 ligase that called NF8. So NF8 make this ubiquination chain to the CHEK2 and lead to the CHEK2 degradation. And at the same time, NF8 has its auto-ubiquination activity. So it's add ubiquitin to the CHEK2, and also it generate a polyubiquinate chain for the NF8. So what we found is ARID1A deficiency lead to the disruption of NF8 and the CHEK2 interaction. So it's kind of without ARID1A, the E3 ligase cannot properly associate it with E3 ligase subject CHEK2. So how does it happen? It's because the loss of ARID1A caused the polyubiquination of NF8. This polyubiquination does not lead to NF8 degradation, but it leads to the dissociation of CHEK2. So maybe we suspect NF8 polyubiquination has all this chain modification. It interrupts interaction between the NF8 and the CHEK2. So in summary, what we identified is in ARID1A deficient cells, we found that this increased CHEK2 expression is likely through the ARID1A regulated ubiquination process that's going to alter the DNA damage checkpoint signaling. So if the DNA damage checkpoint signaling is altered, what is the molecular consequence if we inhibit CHEK2? So here we use the DNA fiber assay to study if we treat the cells with the CHEK2 inhibitor or maybe ATM inhibitor, what's the cellular consequence? So one unique consequence as detected by this fiber assay is the replication fork errors or lesions. So basically we have CIDU, we do the first labeling. This CIDU will be detected by the red fluorescent. Then we treat the cells with the CHEK2 inhibitor. After 2 hours, we wash away the drug and we label the cells with the IDU. So this IDU will incorporate into the DNA, and later we're going to label this IDU with the green fluorescent. So as you can see here, if the cells have a very good DNA replication, it will incorporate the red fluorescent. That's the CIDU incorporated DNA. So if the cells can have a smooth ongoing replication fork, you will have the fiber have also red and green labeling. So that's indicated ongoing replication forks. And if you only see there's a fiber, only have the red labeling, does not have the green labeling, that is the stored replication fork because it's labeled by the red fluorescent. Once we treated the cells, this replication stopped or maybe stored. It never initiated. So that's why it's only labeled by the red fluorescent. And also we have another type of labeling that is only have the green. So that's the new origin labeling. That means this DNA was not synthesized when we have the red labeling. After we treat the cells with CHEK2 and remove the drugs, this is the replication fork started. So we call it a new replication origins. So by quantitate these 3 types of labeling, we can analyze what's the impact of CHEK2 inhibitor on the replication fork progression. As you can see here, we do see in the ARID1A knockdown cells, if we treat the cells with CHEK2 compared to the control parental cells, we do see this reduced ongoing forks. That means the cells cannot continue uptake the green labeling, so we don't have a lot of ongoing forks. And we do see this increased stored forks. That represents the replication fork uptake the red fluorescent labeling, but it stopped. It never reinitiated. And also, we observed the new origin firing was reduced. There was not too much reinitiation after we removed the drugs. This data showed ARID1A deficient tumors are more sensitive to the CHEK2 inhibitors compared to the control cells. And also we use the ATM inhibitor because ATM inhibitor is upstream kinase of CHEK2. It controls in the similar signaling pathway. So if this is a pathway related function on the DNA damage checkpoint, it is naturally, we're going to show very similar result, use the ATM inhibitor. So indeed, we observe very similar phenotype when we use ATM inhibitor. So more interestingly, as you can see, we do have more problems from the replication than when we stain the cells with the pico green. That's the DNA dye that can detect DNA in the cytosol. So as you can see here, because this dye can detect small fragment of DNA. So this is a cytosolic DNA, this is a nuclear stain, and we do see this little, like little dot fluorescent in the cytosolic. So this is the accumulation of cytosolic DNA. And as you can see, compare with our control cells, these are control cells. After treatment with the ATM inhibitor or ATI inhibitor, this is a Cto inhibitor, this is an ATM inhibitor, we do see the increased accumulation of cytosolic DNA in the cells. This is the quantitative data. So if we have this cytosolic DNA accumulation, what is the molecular impact? From the innate immunity studies, it has been demonstrated for a long time when the cells has cytosolic DNA either derived from the virus or from the bacteria, it will activate the innate immune pathways, such as the CGAS STING-mediated innate immune response. So basically, what we identified from our studies, we have a DNA damage that is not from virus or bacteria, actually it's from our own cells genome. Those lead to the accumulation of cytosolic double-stranded DNA, and it gets recognized by the CGAS, the cDNA, double-stranded DNA sensor. And this CGAS has the enzyme activity to transform the ATP and the GTP into the cGAMP. So cGAMP becomes very important signaling molecule that is activate the STING pathway. So the STING activation will cause the phosphorylation of IRF3 and the TPK1. These are the two essential transcription factors. It's going to translocate it into the nucleus after phosphorylation and activate the type 1 interferon response. So this type 1 interferon response provides a very important role for the anti-tumor immunity as well, because type 1 interferon response or maybe innate immune response can prime the cytosolic T cell mediated adaptive immune response. So as I show the pathway here, then we start to conduct experiments to test what's the molecular consequence if we see the accumulation of cytosolic DNA. So first, we did the Western blot. As you can see here, this is our Western blot analysis. We have shRNA knockdown ARID1A. We do see in the presence of, this is a Cto inhibitor, this is ATM inhibitor, there's a strong activation of phospho-TPK1. Without the treatment, we didn't see too much difference. This is correlated with our DNA fiber analysis because Cto inhibitor and ATM inhibitor cause the replication difficulties and errors. So having identified the phospho-TPK1 activation, next we test whether there's increased response to immunocheckpoint blockade by using the DNA damage checkpoint inhibitors. As I mentioned, the type 1 interferon activation could lead to anti-tumor immune response as well. So we choose the ATM inhibitor, as you can see here. This is our parental tumor. Regardless ATM treatment or the anti-PD-L1 treatment, we did not see any significant improve of the mouse survival. So basically, there was not too much efficacy in the treatment effects. But in the ARID1A knockout tumors, we did see the combination with the ATM inhibitor with the anti-PD-L1 antibodies, it improved mice survival. So we did not have a large cohort, but in this small cohort, we did achieve the statistical significance. So this means ATM inhibitor may potentiate the therapeutic efficacy of anti-PD-L1 in ARID1A-deficient tumors through the activation of DNA damage-induced innate immune response. So to validate our mouse work, we went back to the TCGA data. We stratified the patient with the ARID1A mutant status and also ATM mutation status. So these are both wild type tumors. This is double mutant tumors. So compared to the single mutant tumors and also the wild type tumors, the double mutant tumors do show increased TIL score. That means there's increased tumor lymphocytes infiltration. And here is the mRNA level. So we strated the patient tumors based on the ARID1A mRNA expression and ATM mRNA expression. And here is the Cto mRNA and the ARID1A mRNA level, because overall Cto mutation rate was too low to allow us to do any statistical analysis. But when we stratified the patient by the mRNA expression level, we did see the patient with double low, it has better TIL score, like immune response. And also in the patient survival, the double mutant has even better survival outcome compared to ARID1A mutant tumors. So this is our study with the ATM mutant and also the Cto mutant tumors. So this validate what we hypothesized and validated in the tumor data. So this suggests maybe the ATM Cto signaling pathway could be a very specific target for the ARID1A-deficient tumors. So here I want to summarize the ARID1A studies conducted in my lab. I have to acknowledge the very important role of ARID1A or SWI/SNP complex in the transcription regulation. And based on the transcription analysis, a lot of good publications and a very interesting result has been identified how ARID1A control the transcription or maybe the enhancer function to function as a tumor suppressor, and also a variety of inhibitors targeting transcription, such as the EZH2 inhibitor, HDAC inhibitor, has been identified by the people in the field to treat ARID1A deficient tumors. So in my lab or in my ARID1A study, we are completely focused on the DNA damage response and also the genomic instability part of the ARID1A. As I mentioned to you, chromatin remodeling is required for the transcription, the protein access to the DNA. It also required for the replication factors and the DNA repair factors to get access to the DNA. We want to specifically contribute to this area instead of doing the transcription, of course, because I'm a biologist studying the genomic instability mechanism that fit into my research expertise. And also we would like to contribute something different from the crowd of the field. They are focused on the transcription. And by studying the DNA damage response, we identified ARID1A controlled ATR activation that potentially sensitize the patient to the PARP inhibitors. But in the clinical setting, whether the patient with ARID1A mutant can 100% sensitive to PARP inhibitor, maybe the story is not that simple, because as we know, ARID1A is coexisted, mutation of ARID1A coexisted with the PI3 kinase mutation. So we don't know whether this combination mutation is going to change the response. So that's a lot of studies we can expand to test what is the determination additional to the ARID1A deficiency that may rewire the DNA response and change the PARP inhibitor sensitivity. It's like the BRCA1 and the 53BP1 story, because the BRCA1 mutation sensitized to PARP inhibitor. But if the patient has the 53BP1 mutation or loss of 53BP1 on top of BRCA1, it will rescue the DNA repair defect and cause the resistance to PARP inhibitors. So that's called synthetic rescue in addition to synthetic lethality. So that's one potential direction we can pursue in future. And also we identified ARID1A control the mismatch repair pathway. That's another DNA repair pathway, maintain genomic stability. This leads to the anti-PD-L1 treatment sensitivity. And more importantly, beyond the chromatin associated process, our study on the Cto protein ubiquintination indicate a non-chromatin dependent function of ARID1A. because for such a long time, all the ARID1A function is associated with the SWI/SNF complex, is associated with the chromatin remodeling activity. But we do see ARID1A function as a regulator of the ubiquination process. That leads to a new area of study of SWI/SNF and ARID1A. So from this study, we identified targeting ATM or Cto signaling pathway may help us to identify a rational combination to target ARID1A-deficient tumor. So here I want to emphasize the Cto inhibitor in the clinical is not as popular as the CHEK1 inhibitor. Also, ATM inhibitor is not as popular as ATI inhibitor because both inhibitors did not show as promising as the tumor suppression effect compared to the ATI inhibitor or the CHEK1 inhibitor. And indeed, in the mouse model study, CHEK1 knockout mice is umbilical lethal. The mice cannot survive. But the Cto knockout mice have relative normal phenotype. So they argue Cto.
I'm sorry to interrupt, but we are past our 2 o'clock time. So I'm just gonna ask if maybe we can wind down just to be respectful of everyone's time.
This is my last slide. Yeah. Hello. Yeah. So this I want to just emphasize one point about the DDR inhibitors. It's not only just to suppress tumor. Maybe as we showed here, it can be used as immunomodulation agents in a context-dependent pathway such as ARID1A deficiency. So this is the overall of the project we are doing with ARID1A, and we expect we can conduct our additional new experiment to translate what we identified of ARID1A in genomic stability control, extend our research to the cholangiocarcinoma. That's really our hope for the next step. And we're currently conducting experiment in the new models to study how we can improve the treatment for the patients. Yeah, this is actually my last slide. So I want to thank everybody for attending the virtual meeting, and I look forward to any questions.
Thank you so much, Dr. Peng. That was a whirlwind tour through DNA damage and ARID1A, and thank you very much for all of your work. I think, unfortunately, we may not have time for questions today, but we will make Dr. Peng's contact information available if anyone has any questions in the future, yeah?
Yeah, sure. Anyone, just send me email or anything. If there's collaboration opportunities, I will really appreciate that, because for this cancer type, I'm a newcomer, but for the biological pathway, I do feel because the clinical significance of ARID1A, the mutation in this cancer type, we probably can contribute to some unique angle to improve the clinical outcome.
Well, that's always the mission, so let's hope so. Everyone, we want to wish you a happy holidays from ICRN, and have a wonderful holiday season and new year, and we will see you for the seminar series in January. Those emails will go out soon, so have a great few weeks. Bye-bye.
Bye-bye.